Welcome everyone to our second session of Headwaters Science Institute Research Experience Program. We have many students this summer who have worked very hard to go from a question that they've had from a topic of interest to forming a hypothesis, to collecting data on their topic of interest, adjusting their methods when needed, analyzing their data, discussing their results, and coming to conclusions. And today we're very excited to share the presentations from these projects with you. My name is Leslie McGinnis, or Dr. Leslie McGinnis, and I was the primary teacher for this summer's class. And we are delighted to be able to share this second session of students and their projects with you. And without further ado, I'm going to add the first student and their mentor for brief introductions of the student. So I'd like to introduce Marin to introduce our first student of this session.
Yes, hello. My name is Marin. I am an environmental chemist in the world of microplastics. I work at the Desert Research Institute in Reno, Nevada. And of course, I have had the immense pleasure of being Kino's mentor this summer, although I will say about him, he doesn't need much mentoring. He's an incredibly bright student. He's from San Diego, and I have truly loved working with him. He is one of the most brilliant kids I've worked with in his age group, although he would never probably toot his horn to that degree. But he has an amazing, relevant personal project, and I'm extraordinarily proud of him. So without further introduction, Kino.
Yeah, my name is Kino, and this is my presentation. So I did mine about hot tubs and the hidden bacteria inside of them, specifically focusing on the risk of Pseudomonas in hot tubs. And I compared like hot tubs connected to pools to hot tubs that aren't connected to pools, kind of isolated ones. Pseudomonas is a common bacterium that thrives in water and moist environments, even under low nutrient conditions or in the presence of disinfectants for healthy people. It's usually harmless, but it poses a serious risk to individuals with weakened immune systems or chronic lung conditions like cystic fibrosis. In people with CF, this bacterium can cause persistent lung infections, leading to chronic inflammation and long-term lung damage. Hot tubs are a known source of Pseudomonas, especially when sanitation isn't maintained well. The warm, moist environment makes them ideal for bacterial growth, while larger, pool-connected systems often have stronger filtration and chemical control. Isolated closed system hot tubs may be riskier, though this isn't fully understood yet. So while hot tubs may seem relaxing for some, they can pose an invisible health risk if not properly contained for others. So to test this hypothesis, I collected water samples from a total of 8 different hot tubs. 4 of these hot tubs were isolated systems, meaning they were not connected to any pool or other water system. The other 4 were hot tubs connected to larger pools, which typically have continuous water circulation and filtration. I used the AquaVal Pseudomonas test kits, which are designed to detect the presence of Pseudomonas in water. For each hot tub, I followed the instructions provided with the test kit to collect a sample and observe any color changes indicating bacterial contamination. All tests were conducted during the same week to limit variability due to temperature or weather, and I recorded whether each sample tested positive or negative for Pseudomonas and performed a chi-square analysis. So out of the 8 hot tubs tested, 3 out of 4 isolated hot tubs tested positive for Pseudomonas, while only 1 out of the 4 connected, while in 1 of the 4 connected hot tubs tested positive. This indicates a higher risk of contamination in isolated systems compared to those integrated with larger pool filtration systems. The difference in contamination rates suggests a relationship between the type of hot tub system and the presence of Pseudomonas. So my p-value for my chi-square test was 0.0285, and oh wait, I need to switch slides. It was a small sample size. The results support the hypothesis that hot tubs which are connected to pools have higher rates of contamination than isolated hot tubs. So I used the chi-square analysis, and so I used it to compare the number of contaminated hot tubs between isolated and connected systems. This test helps determine whether the difference in contamination rates is likely due to chance or if it's statistically significant. Basically, it compares the result I observe with what we would expect if there were no real difference between the two groups. In this case, the p-value is 0.0285, which means there's a low probability that the difference happened by chance, suggesting a real link between the type of system and contamination. Our findings indicate that isolated hot tubs present a significantly higher risk of Pseudomonas contamination, with a p-value of 0.0285, making the results statistically significant. This supports our original hypothesis and matches what's already known about Pseudomonas, or about Pseudomonas and how it thrives in warm, moist environments with poor sanitation. One key factor here is system design. Isolated hot tubs often have less frequent maintenance, inconsistent chlorine levels, and limited water circulation. In contrast, hot tubs connected to pools tend to benefit from better filtration and chemical monitoring. This design difference could explain the higher contamination rates in isolated systems. This has important health implications, especially for individuals with cystic fibrosis or compromised immune systems. For them, choosing hot tubs that are connected to larger, well-maintained pools may reduce infection risk. That said, there are some limitations to the study. Sample size was small, and we couldn't control for key variables like usage patterns, hot tub age, or cleaning routines. Also, while the Aquaval test confirmed the presence of Pseudomonas, it did not measure like how much of it was actually there. So it only detected presence. Despite those limitations, this research offers a solid starting point. Future studies should aim for larger sample size and controlled testing environments. In the meantime, our recommendation is that at-risk individuals avoid isolated hot tubs and choose systems with more consistent regulation. Future limitations or future directions. Looking ahead, there are several ways this research can be expanded to build stronger evidence. First, increasing the sample size would help improve statistical power and reliability. A larger data set would also allow us to detect patterns that weren't visible in the small pilot study. Future studies should also include more controlled variables, factors that I've already stated, like the age of the hot tub, how often it's used, and how it's maintained, can all influence bacterial growth, and we need to account for those. In terms of testing, using lab-based methods would be a major improvement rather than just detecting the presence of Pseudomonas. Future work could measure the actual bacterial concentrations and even identify specific strains, some of which may be more harmful than others. It would also be useful to study the water chemistry itself, such as chlorine levels and pH, to better understand how these affect bacterial survival and growth. Finally, future research could aim to establish clear public health guidelines, especially for vulnerable populations like people with cystic fibrosis. If we can identify which types of systems are safer, we can help those individuals make more informed decisions about hot tub use. Any questions?
Yay! So I'm going to bring on your mentor as well for any questions. Okay, I'm checking the chat to see if we have any questions. So I'll ask a question. After doing this experiment, what recommendations would you have for people that are immune compromised, not just with CF, but other types of immune compromised, other immune compromised conditions?
Yeah. So I would say that if anybody with a compromisation that relates to like lungs or anything that could put aerosolized dangerous bacteria that could put them at risk, I would say stay away from basically any isolated hot tub, and just to be safe, maybe all hot tubs, because there is still risk going into hot tubs that are well circulated. So yeah, because it could be bad if you get that.
All right, great job. Thank you.
Thank you.
Okay, and I'll add on our next mentor and student. I'd like to introduce Dr. Jesse Scott.
Hi, my name is Jesse Scott, and I am proud to be Mina's mentor during this semester. She has done a fantastic job of assessing this incredible research project, and the implications that are discussed, that Mina will be discussing, will have some long-term effect. And there's definitely room for future research. And so I'm excited to share this work that Mina has been doing. Thank you.
Wonderful. And I will take myself off and Jesse, just a moment. Okay, Mina, it's all yours.
Okay, I'm Mina Nandela, and my project was understanding the correlation of ocean acidification to kelp area after an El Nino event. Sorry. Okay, so a little bit of background. I decided to choose this project because I was really interested in ocean acidification, and I knew how it affected calcifying organisms, but not so much other ones, so I decided to focus on kelp. So for some background information, ocean acidification is a phenomenon where CO2 is absorbed from the atmosphere and causing the ocean to lower its pH and become more acidic. And previous studies have shown that its effects on kelp didn't cause a significant change in the photosynthesis process or the amount of kelp. And El Nino is a climate pattern that warms waters, and a previous study shows that it significantly reduced the amount of kelp. Okay, so this introduces to our research question, which was, since the disturbance event in 2014, the El Nino event, what are the effects of ocean acidification on kelp abundance in Southern California in the years 2015 to 2021? So I hypothesized, based on the previous information, that as ocean acidification occurs, lowering the pH, the kelp abundance in Southern California will be diminished significantly. Okay, so I conducted this study using two data sets from assessing kelp from Kelp Watch and ocean acidification and a bunch of variables from the California Cooperative Oceanic Fisheries Investigation. And I constrained both these data sets by both the location of Southern California, so they would match up with each other, and the time frame of 2015 to 2021, so after the 2014 event. And then I combined these into one data set so I could use R analysis and use statistical analysis. So one of the first things that I did was create a correlation matrix, and this basically shows which variables are more or less related to each other. So based on this matrix, I in the following slides used mean DIC, or dissolved inorganic carbon, and mean temperature and mean kelp area. Okay, so using a time series analysis, we were able to see that there's a seasonal variation in kelp every year, and whereas dissolved inorganic carbon and temperature are more consistent. And basically what the seasonal trend is increasing during quarters 2 and 3, which is spring and summer, and decreasing in quarters 1 and 4, which is fall and winter. Okay, so for this following slides, we used a time-lagged model, and basically this will show the effects of kelp in 3 months after each point, because the kelp had delayed reactions to the increases in dissolved organic carbon and temperature. So as I said in the methods, we used that matrix to focus on the predictor variables of temperature and dissolved inorganic carbon. And we use these as predictor variables for future kelp area. And as you can tell, this model was statistically significant. And most of the data falls within a standard deviation. And this means that these two variables of dissolved inorganic carbon and temperature explain 46% in the variance of the kelp's response, basically the reduction in kelp compared to the typical kelp growth. And this is similar, it's basically the same graph, but in a different form. And you can tell that this model definitely was significant, and using these two variables explains a good amount of the variance. Okay, so next we conducted a t-test between the mean of the kelp area in the high DIC group and the low DIC group. The one pictured here is actually from the model before we lagged it. So as you can tell, it wasn't significant. But after we time lagged the model, we were able to find that kelp canopy area was significantly lower under high DIC conditions, and kelp area trended lower under high temperature conditions as well. So what this means is that there's a synergistic role between higher dissolved inorganic carbon and higher temperature, which is causing the kelp to reduce in abundance. And so based on the data, we were able to see that there was a combined force between dissolved inorganic carbon and temperature that causes kelp to reduce. So we decided to focus on each variable individually. And as you can tell, these results weren't very significant. Using mean DIC as a predictor variable only explained 1.5% of the data, while only using temperature only explained 14% of the data. What we got from this is that these variables by itself couldn't accurately predict future kelp area, but a combined model using both of them more accurately predicts kelp coverage. Okay, so basically by using a quarterly time series data set and lagged linear regression models, we were able to find that increases in dissolved inorganic carbon and temperature together predicted significant reductions in kelp canopy area in the following season. And these results support the earlier hypothesis that environmental stressors associated with ocean acidification and warming have measurable impacts on coastal kelp ecosystems. And this actually aligns with previous findings that found that elevated temperature limits kelp growth by pushing thermal tolerance thresholds and reducing nutrient availability, and higher dissolved inorganic carbon levels, which is an indicator of ocean acidification, impairs calcifying organisms and disrupts food webs, which indirectly affects kelp health. So some biases that we had during the study is that it was constrained to the area of Southern California. So data gathered in other parts of California or other parts of the world would vary. And we can only use data from the specific points that were measured. So of course, it might fluctuate if measured in other places. So some things to consider for the future is the importance of time-lagged models, especially when evaluating coastal ecosystems, because as you can see, our lag model was able to find significant correlations that our non-lag model wasn't able to find. And then some other questions that would be important to consider in the future is what other factors related to kelp growth, for example, nutrient availability, and how would change in this variable affect kelp over time? And also, how do other climate patterns and disturbance events like El Nino impact kelp area? And then finally, I just want to thank my mentor, Jesse, for guiding me through this research project. And I learned a lot on how to analyze and interpret the data. And then also Leslie and Megan for giving me feedback throughout this process. Thank you.
Yay! Great job. Thank you so much. And the comment section was really happening when you were presenting as well. So I'm going to add on your mentor, but I think you might have some questions before my comment in the audience.
Okay.
Outstanding work, Mina. You did a wonderful job synthesizing this data. For everyone else, as you heard, the implications of understanding this time series and the lag data and using different predictor variables combined can be used to really predict where the direction of that kelp growth will be in the future. And I just thought it was amazing that we were able to predict with so much accuracy what this abundance or the kelp coverage, actually canopy coverage, would be 3 months into the future. So considering all of those different factors that we assessed, if you had the time and the resources to be able to collect data on your own, do you think that would better support your hypothesis and future research?
Yes, I think it would. I think using a lab to be able to test each of these variables individually and how they impact kelp would really strengthen my results, because as of right now, there's a bunch of different factors in the oceans affecting our results. So we can only really tie a correlation to it, but it would be interesting to see how those individual factors affect kelp.
Great. Thank you so much.
Thank you.
Outstanding work.
Okay, wonderful. Do you want to take any questions from the audience?
Sure.
I saw one in here about, well, you can scroll through, see if there's any.
I actually can't see any of
okay. There's one that says, what are factors that affect ocean acidification?
Okay, so in the ocean acidification data, there was a bunch of different factors, which was DIC, temperature, salinity, total alkalinity. Yeah, I think those were the main ones. And then it was just like from where they were from. Yeah.
Okay, wonderful. Thank you so much. And thank you, Jesse, as well. And I'm going to add Jesse on the screen again to introduce our next student. Niket. And let's see. Niket's there.
Hello. Thank you. Can you hear me all right?
Yes.
Yes.
Yeah.
Okay, all right.
All right. Okay, well, let me introduce you real quick. My name is Jesse Scott, if you didn't catch that already. Niket is going to be presenting his research tonight. It's a very interesting research, and it also has quite a few implications, not necessarily oceanographic data, but data that can help support terrestrial biological habitats. So looking forward to hearing that tonight. Thank you.
And thank you, Jesse. And okay, Niket, it's all yours.
Okay. My name is Niket Ramamuti, and this is my presentation, Vegetation Matters, How Having Biodiverse Habitat Sustains Bee Communities. So, first of all, urban green spaces are vital habitats for pollinators within developed areas with scarce natural ecosystems. Some examples are community parks, green medians, residential lawns, and many species of bees are keystone species in these ecosystems due to their critical role in pollination, which supports upwards of 85% of all flowering plants. Urbanization is often a disruptor of this, building over natural landscapes, sprawling over the land, and introducing pollution to the environment. So this study will explore how urban green spaces with different levels of vegetation and plant biodiversity influence these bee populations. So this is the literature I used for my study. It's a lot. So the first article is about the pollinator effect on food, which details how pollinators boost crop variety, yield, and quality. The second article is about the pollinator effect on the ecosystem, about how having diverse pollinator communities stabilize ecosystems and promote this resilience. And the third is the most important. It's about the effect of urbanization on pollinators. And it details how these highly urbanized areas see low pollinator diversity and how you really see bee populations thrive in these urban green spaces with native plant richness, spontaneous flora. So my hypothesis is that urban green spaces with dense, healthy, biodiverse vegetation sustains a greater abundance of bee population than spaces with sparse, low biodiversity vegetation. And it's expected that we'll see sites that have more native plants and dense understory layer would have greater bee species biodiversity and abundance. So to conduct this study, we chose 2 hillside plots, 7 meters by 7 meters, around 100 feet from residential areas, both fully exposed to the sun with consistent light, elevation, and surrounding foliage. These 2 sites are completely identical except for whether they have native or invasive flowers and whether they have dense or weak chaparral. And this is what distinguishes between a site with biodiversity and a site without biodiversity. So site A is the dense native grass, native wildflower site, indicating strong biodiversity. Site B is lacking understory, covered in invasive plants, low biodiversity. So to sample this, first we analyzed both of the habitats. So we checked native versus non-native flowers and also understory density by calculating average understory height times estimate percent cover. And then to catalog the bees, we conducted 12 15-minute observation periods during peak activity hours, and we wanted to count bee abundance. So this is number of bees observed. Bee diversity, where we catalog bees based on morphotypes. So big black bees, small orange bees, small black bees. And we did 6 visits per site just to have a large pool of data to select from. And the results of this, we saw that Site A had a significantly higher bee count than Site B. All types of bees were more abundant at Site A. And so we counted a total of 108 bees across 12 site visits conducted between July 9th and July 15th with equal sampling at the 2 locations. So observations included 3 distinct bee morphotypes: big black, small orange, small black, and bee abundance varied notably. So in the graph here, graphs for each morphotype and total bee counts for both of the sites are shown. Then to calculate bee abundance, we used a box plot map, and we saw that Site A showed a much higher average bee count per site than did Site B. So both the average, which is represented by the black diamond, and the median, which is represented by the horizontal black line, were higher for Morgan than for Lewis. Were higher for site A than site B. And this box plot represents total bee abundance again, where the shaded portion of each box plot is the interquartile range, 50% of the data. And again, just indicating that there were more individuals in site A than site B. And we did a linear regression map just to check if there was a correlation between temperature and bee abundance. And we found no significant correlation between temperature and bee population. There was an R value of 0.22, so a slight decrease, but a P value of 0.49. And it was too weak to be relevant, and it showed that the temperature didn't skew the results of our data. And again, this is a graph of the regression plot of bee abundance to site temperature. So from our results, we found that site A, which had dense understory and native flowers, attracted significantly larger amount of bees than site B did, which had invasive flowers but no wild grasses. These findings indicate a strong site-specific effect on bee abundance linked to differences in floral diversity, plant composition, and proximity to urban areas. And additional sampling across different times of the year, different vegetation surveys of the habitats would help us maybe more closely identify the possible environmental variables behind these findings. The results of this study match with other literature on pollinators. For example, the Paris Urban Ecosystem Studies found that impervious surfaces and loss of vegetative complexity lower pollinator populations. And it also supports the idea that pollinator health and ecosystem productivity are directly correlated. So some limitations of this study: we had a small sample size. We had only 2 sites, 6 visits per site, which limits the statistical power and interpretations of the analysis, and also limited materials because we had to classify bee by morphological features instead of specific species. Also, there was possible observer bias due to only visual serving. Fast-moving bees could be easily missed. So the conclusions from this data, it supports my hypothesis that native plant diversity and dense understory vegetation increases pollinator abundance. And these 2 sites are just very striking examples of different approaches to urban green space design. And we can see that bees thrive in spaces with rich native biodiversity. And bees flourishing within our cities strengthens pollinator connections and vegetation, even in extremely urbanized spaces. Having dense, biodiverse green space creates a habitat for pollinators to flourish in very impervious green urban spaces. And that's my presentation. Thank you for listening. If you have any questions, please put it in the chat.
Yay! I'm going to add your mentor on. And because I am an entomologist, I would love to ask, what was the most adorable or most awesome looking bee that you saw in this area? And I'm asking in part because Headwaters had a San Ramon camp last week, and we saw a lot of really cool bugs. So I'm curious, which is your favorite?
Yeah, I think for me, my favorite was what we called the big black bee. I don't know what species specifically, but it was like this, like very fuzzy, black, large thing just like flying through the air. It was really cool to see. There was a lot of them too in Site A. So
cool. Let's see if we have any questions.
I have one question. Great work, Niket. You did a good job of synthesizing this data. There's some interesting observations that you had, and you did a great job kind of correlating this research with other peer-reviewed literature, and you did a great job of explaining your research in the discussion. Do you think in the future, if we were to conduct another study, do you think there would be a different or a better way to identify those bee species next time?
I think.
How would you do that?
I think, yeah, there will be a lot of more accurate methods to do in a maybe more fleshed out study. I would say catching bees with a net and like analyzing them specifically would be a lot more helpful if you wanted to see data pertaining to specific species of bees. Like, of course, morphotypes using um, um, observations still displays that bee diversity well, but if you want to see per species how each species is affected, it will be good to use tools like a net to just analyze it specifically.
Thank you.
Yeah, and I believe we talked about this a little bit and some of the resources to identify them. And it can be pretty challenging, especially when things are moving fast and you're trying to use even specialized bee and butterfly binoculars. So it does take a lot of work. And I was seeing in comments, AI, doesn't tend to do as good a job as you might think. So, but thank you so much, Niket. And another round. Okay. And I'm going to get ready the next person. Okay, so the next student, Ariana Banerjee, was my student this summer. And so we have another insect themed presentation right after a bee presentation. And I wanted to say that throughout the time working with Ariana, I was struck by her curiosity about the world around her and her drive to understand the social systems and behaviors of insects, and that she needed very little direction and enjoyed trying out methods herself. And I had a really great time talking about some of the ways that we could represent different options to ants and then what some of the confounding variables would be. And without further ado, I will add her presentation so she can tell you more about it.
So, hi, my name is Ariana Banerjee, and my study is on the impact of color vision on ant foraging behavior. I chose to research ants because I have a colony of Argentine ants that actually live in my backyard, which kind of piqued my interest in these insects. I wanted to learn more about their behavior and understand why they choose to eat the food that they do. So, ants typically rely on chemoreception, which is the complex sense of smell and taste to find food and explore their environment. While the reliance on these senses are pretty well documented, recent studies have shown that vision, especially with color, can also play a role in their behavior. Color has also been shown to influence foraging in pollinators and other insects, but its influence on ants is not that well known. Studying this could also contribute to a better understanding of ant sensory and decision making. Previous studies on ant vision say that many species have limited color perception, which is often restricted to a narrow range of wavelengths. This range typically is of UV to green, which is around 300 to 650 nanometers. However, research has shown that ants can learn to associate certain colors with food, meaning that color could affect their behavior in certain situations. Understanding whether ants respond differently to specific colors can also be used for pest control and ecological studies. So, in order to learn about whether ants have a preference for certain colors when searching for food, I conducted this study. And on this slide, I included a picture of the ant that was in my experiment, which is the Argentine ant. They're, I believe, pretty common in California. So for my experiment, the research question that I went with is, how does color impact ant foraging and food preferences? And for my hypothesis, since I didn't have enough information on color choices to pick a specific one, I decided to go with the null, which is that the ants have no preference towards any color. To conduct this experiment, the main material I used were 7 by 7 inch colored plates. The colors selected for testing were red, yellow, blue, and white, with white being the control. These were chosen to include a range of varying colors. Next, I made sugar water by mixing white granulated sugar with tap water and stirred them together until fully dissolved. For each plate, I poured and measured about a half tablespoon of the sugar water onto the center of each plate to be served as bait. At each testing site, the 4 plates were placed on the ground in a square arrangement, which you can see in the picture, and there were about 1 to 2 inches of distance between each other. The plates were positioned on a flat open ground where ants were already active. I started every experiment at around 6 p.m. because I previously tested this, and I observed that ants in my area were most active around then. I conducted all of my tests within 3 days so that I could reduce the variation in weather and temperature. Once the plates were set, I set a timer for an hour and a half. Once the time ended, I counted the number of ants present on each plate. This process was repeated at 4 different locations so I can increase the environmental variation and also reduce any location bias. The locations were my backyard, a sidewalk pavement, and 2 different parks. The picture here was in my backyard. And here's some other pictures. The one on the left is the pavement, and the one on the right is another park I went to. For the data I collected, I conducted an ANOVA test to compare my 4 groups and see if there's a statistically significant difference among them. Each data point represents one site. The result of this was a p-value of 0.13, which means it's statistically insignificant but shows that there could be a difference. My sample size was quite small, so a larger one may yield a change in the significance. Each group has a varying number of ants, but it's clear that the white one had one site with the highest number of ants at 30, which could have skewed the analysis a bit. And after the ANOVA test, I conducted a post-hoc Tukey test, which compared all of the possible pairs of colors. The table shows those comparisons. As said in the interpretation of P column, all of them have a very low indication or no evidence that there is any difference in the groups, making it statistically insignificant again. However, the p-value of the red and white plates show that there is possibly a difference between them. A common trend I noticed with all of these is that the pairings with white seem to be more significant than the ones that have only other colors. So, in conclusion, both the ANOVA and post-hoc Tukey test resulted in low p-value scores and little indication of any difference in the groups. It indicates that there's no statistical significance in the data. This means that we can fail to reject the null hypothesis. With all of this analysis, my experiment concludes that there's no significant correlation between color and ant foraging behavior or preferences. However, there's also a pattern of ants being more prominent in the white plates rather than colored ones, and that could be something that I could explore more in the future to understand why these ants picked it. There are also a few possible biases that may have impacted the data collection process and eventually affect the test results. One of these would be the location, because I picked experimental sites that were close to my proximity, so all the data would relate to my locality and may not be the same in further areas. For more variety, a different thing I could do is go to further locations. And the other issue was the sample size, because mine was relatively small, so the data wasn't as varied or significant as it could have been with a larger sample size. And the third one is the arrangement of plates. The order that they're placed in could affect how close ants are to one compared to the other. For example, the red plate was closer to the white than blue, which may influence ants on the red to avoid the blue plate. To prevent this from happening, I may want to test out different arrangements and record those results as well. So in conclusion, while the results don't show significance, this experiment is still valuable in showing what certain Argentine ant behavior is like when it comes to foraging with color preference. As mentioned before, this information could also potentially be used in future studies and definitely taught us more about their cognitive skills. Lastly, I would like to thank my mentor Leslie for being an amazing guide and helping me to write my manuscript, plan the entire experiment, and assist with the data analysis. I'd also like to thank my parents who bought all the necessary supplies and assisted me with data collection. Thank you.
Yay! So I'm looking to see if there are any questions in the chat. So one thing I will ask, so Ariana, can you remind me what the patterns that you noticed when you first encountered this ant invasion in your house that later led you to want to do this experiment?
Yeah, so basically, it kind of started out with we kind of just had some, we usually kept food out that we didn't really need space in the cupboard or the fridge. And the ants somehow found this, and then they started going into our house, and it became a whole infestation problem. So then what my parents did, we kind of eventually searched around and found the source was from my backyard. So we did remove the ones in my house, but the main colony in my backyard stuck around. So that's really got my interest.
Okay. And what made you decide to use plates for color versus using like food coloring or dyes?
Yeah, I chose plates because food coloring dyes, the food color itself could have chemicals that may affect how ants perceive that food. And to reduce any of those kind of issues, I decided to go with just the colored plate where I can, control more of the chemicals and any other thing that are used in that.
And I know it was that you primarily saw Argentine ants. Did you see any other species of ants? And do you think it might be different if you sampled in a place that had more native species of ants?
From my studies and the ants that I identified, I only did see Argentine ants. But I do believe with other ant colonies, there could potentially be a difference. Yeah, maybe more urban areas would prefer these ones, but maybe more rural ants would prefer different colors depending on their environment.
Okay. Great. Thank you so much. Great job again. And our next student, a mentor, cannot be here tonight, so I'm going to do the introduction. And Kshitij, do you have your slides up?
Yeah, I'll present them.
Okay. So I have a really wonderful note that was written by Kshitij's mentor that I want to read. If my other computer will cooperate. Okay. So Kshitij's mentor was Aruna Nayagam, and this is the introduction she wrote. Kshitij is a dedicated and committed student whose curiosity and passion for science have truly impressed me. His commitment to his project has been inspiring. And one particularly remarkable aspect was his initiative to replicate a BSL-1 laboratory setup at home using proper sterilization and decontamination techniques. Demonstrating remarkable creativity, he even designed a makeshift incubator using curd tubs and a lamp. Throughout the project, Shitij's creativity, resourcefulness, and consistent engagement stood out as his greatest strengths. He independently sourced and managed all the necessary supplies, followed up on project needs, and even began drafting a research paper with a clear and logical flow. I can clearly see the makings of a young scientist in him, one whose passion will continue to grow well beyond the summer camp. His keen interest in understanding antibiotic resistance led us to formulate a compelling research question: are real-world antibiotics truly controlling bacteria, or are they contributing to resistance? I am excited to see Shitij present his work and feel proud to have mentored him throughout this summer, and I wish him the very best in his future scientific endeavors. So with that, very, very wonderful introduction. I am putting Shitij back up here on the stage.
Hi, yeah. So, the title of my research project is The Increasing Prevalence of Antibiotic Resistance, and it's by me. I'm a rising junior at Basis Independent Fremont in California, and my mentor, Miss Aruna Nagayam. So, just a quick introduction about what exactly are antibiotics, because the term is thrown around a lot. But antibiotics are basically substances that either inhibit growth of microorganisms or destroy and kill microorganisms themselves. So, a little bit about the history. So, Alexander Fleming was the first to discover antibiotics, and he accidentally did by growing a culture of Staphylococcus aureus. Um, and then he just let it grow for about 3 weeks. When he came back, he realized that it was contaminated with fungi, and the area around the fungi, he realized that there was no bacterial growth there. And further research showed that the fungi, which was called penicillium, which is from the genus Penicillium, produced substances that inhibited the growth of microorganisms, and the antibiotic was born. So, antibiotic resistance has become a problem for a while now. It's been around since the 40s. So, just not even 15 years after the discovery of penicillin, a few E. coli strains started showing resistance. The resistance for penicillin was temporarily stopped with the introduction of methicillin, which is what's termed a second generation antibiotic, and it was created in 1959. But just 2 years after that, in 1961, the first cases of MRSA, which is known as methicillin-resistant Staphylococcus aureus, were reported in the UK. And by the late 1960s, a majority of Staphylococcus aureus strains were resistant to penicillin. And by the 1970s, you saw other diseases like pneumonia also becoming resistant to penicillin. So the two main factors that are listed a lot for increasing antibiotic resistance are the increased availability and usage of antibiotics. So basically, people popping antibiotics for anything as if they feel sick, even though it might not even be caused by a bacteria, and the agricultural use of antibiotics. So pesticides and herbicides, they can cause antibiotic resistance in species that infect plants. So my question, looking at this observation, was what is the effect of a continued exposure to antibiotics on the prevalence of antibiotic resistance in a bacterial population? And this involves a little bit of ecology. So the whole thing about natural selection, antibiotics basically act as a selector. They're part of the environment, and so they favor the bacteria who are resistant to that. And resistant bacteria have a variety of ways to be resistant, like making certain molecules that deactivate antibiotics. And so these antibiotics serve to give resistant bacteria an advantage, and killing off all the other bacteria, and that leads to the resistant bacteria starting to reproduce more and more than other bacteria, and over time you'll get the evolution of a bacteria population that is much more resistant than the original population. And since bacteria can reproduce so quickly, this happens in a few years, and nowadays we're seeing bacteria getting resistant in months or even weeks. So for the experiment, the materials I used are listed here. Some important materials. So the water samples from several different lake locations. I used 3 lakes in this case: Lake Elizabeth, Quarry Lake, and a lake a few hours away from the Bay Area to get some more diversity. But I used these water samples mainly because the safety concerns of transporting E. coli from a lab without the proper facilities and infrastructure would just it wouldn't be safe, and me and my mentor did not want to do that. So and the rest of the materials are pretty like self-explanatory. The incubator, though, I, like most people, don't have access to like a $1,000 incubator. So I tried making my own out of 2 tubs of curd or Greek yogurt, and these materials right here. And I'll talk more about how I made them in the methods section, which is up next. So the methods on the left, this is for only the antibiotic resistant test, which is called a Kirby-Bohr test in microbiology. But that's the only thing that is here. There are other procedures that were done, but in the interest of time, I'm skipping over them. And on the right is how I made my incubator. And this is the final picture of the incubator. So bacteria sit in this space between the 2 tubs of curd, and the light shines through here, allowing the heat to rise and heat up the bacteria and keep the temperature at an optimal area. So the whole methods of the experiment was basically to measure the ZOI, or zone of inhibition, and basically that's done through growing a bacterial lawn and then putting antibiotic discs on it, and the area around the discs where there's no bacterial growth, that would be the zone of inhibition, and that basically shows that the bacteria have not grown there, and that shows that they're not resistant past that. So smaller zone of inhibition means a larger frequency of antibiotic resistant bacteria. And this is just the techniques I use. So the bacterial lawn technique is on the left, and the streak plate method, which was used to isolate the E. coli colonies from the water samples, is on the right here. So here are the results from trials 1 and 2. And I just like to note that the experiment is still ongoing. So I will have more trials done even after this. So trial number 1, I used 2 plates. And you can clearly see in plate number 1 that there is a clear zone of inhibition. So this circle that's clearly separated from the rest of the plate, those 2 circles are the zones of inhibition. And they measure about 1.5 centimeters across. And then the radius is 1.5 centimeters. So they're 3 centimeters across. And then in trial number 2, we only got one really good plate number 1. So plates 2 through 4, they might, the indication of no zone of inhibition says 2 things, or might say 2 things: either that the bacteria here have become completely resistant, or that there was just no growth and due to human error. But this plate number 1 here shows 2 zones of inhibition, but they are slightly touching each other, so that does complicate measurements. So the radius, I measured the length from one area across. So this whole thing was 4.4 centimeters, And to get the radius, I just divide that by 4. So yeah, these are the results. And again, more results will come soon. So yeah. But you can see here clearly that the zones of inhibition did decrease for plate number 1. And that does mean that the bacteria did evolve some resistance. So moving on to the discussion. So basically, the experiment does show that antibiotics might do have a tendency to make bacteria resistant, and it's only going to increase due to a variety of factors like increased availability and the larger use of antibiotics in agriculture, because there's just more demand for food and it's easier to use antibiotics to increase crop yield. And here are just some graphs to prove that point. So from about 1960 to 2010, there was a massive increase in the amount of enzymes that antibiotic-resistant bacteria have. And in the graph on the right, there was also a massive increase in the amount of antibiotic usage in crops. And increased usage, as we've already established, leads to increased resistance. So the conclusion I drew from this is that antibiotics might lose their importance in the decades to come as bacteria evolve more resistance, and we as humans can't keep making more antibiotics faster than bacteria become resistant to them. And the hypothesis does seem to be at least partially supported, because ZOIs are decreasing in size in some of the trials. Potential sources of error, human error was a big factor, but also the factor that while the incubator was very successful, it still can't replicate exactly the optimal temperatures that other incubators, other more expensive ones, can. So that could also be a source for potential error. And some further research for antibiotic resistance. These 3, phage therapy, preventative vaccination, and antimicrobial peptides, have shown very large amounts of success, especially phage therapy. So bacteriophages, as they're known, are just viruses that infect bacteria. So they have shown to be much more effective in not only killing bacterial infections, but also stopping bacterial resistance to antibiotics. I'd like to acknowledge Ms. Aruna Nagayam. She helped a lot throughout the whole research project. Also, my parents for just handling the logistics of this whole experiment. And also, of course, to Leslie, Megan, Joyce, and everyone else at Headwaters who make this research experience possible for students like me. And these are my references.
Yay! So I had a question that I wanted to ask you.
Yeah.
And I put it in the chat. So sometimes I think people think of science as something that can be difficult to access, something that can only happen in labs with expensive equipment. But what have you learned in your experience here, making your own do-it-yourself engineering to make science, happen and work out for your project? And how do you think it connects to making science more available to other people?
Yeah, so the fact that I could even have an incubator that remotely keeps the temperature in the optimal range for growing bacteria, made me very happy, of course, because it was so satisfying to see that like the temperature stayed constant, and I actually got a few good plates, and it definitely shows that it's possible. You don't have to have like massive amounts of money and equipment to do it, and yeah, it's just encouraging for anyone else who wants to try these types of experiments.
Okay, awesome. Are there any other questions in the audience? double checking the chat. And I might have missed this, but what made you decide to use local lakes as a place to get water samples?
Oh yeah, mainly because of safety concerns. It would have been hard to transport bacteria, um, like across like several hours, and because that's where my mentor's lab is, and it would be hard for us to like meet up and safely have the bacteria and move it from her lab to my house. So yeah, that's why I decided to get it from local lakes instead.
Okay, super awesome. All right, thank you so much. And I'm going to get our next student and mentor ready. Okay, and I'd like to introduce Marin again.
Hi, nice to see everyone again. My pleasure again to introduce Kushi. I had the immense pleasure of being able to meet Kushi in person at a camp this summer, and I can tell all of you firsthand that she is an extremely bright young scientist who, amongst probably one of the busiest summers of her life, put so much time and effort into this project and managed to stump me a good handful of times by choosing something very data intensive, but of course very relevant and very personal. So very proud of you, Kushi, and take it away.
And it's all yours, Kushi.
Hi, my name is Kushi Gowda, and I'm a 9th grader at Redlands High School. With assistance from my mentor, Marin Monteith, we put together the Examining Substance Abuse and Poverty Markers as Related to Mental Health, a Profile of Modern America presentation. Introduction. The factors that I chose for this presentation were mental health, stress, substance abuse, and poverty. Mental health affects thoughts, emotions, relationships, and daily functioning. It's shaped by life experiences and social environments. Stress can lead to emotional and physical health problems, tying it to mental health. Social stressors, which can include isolation, bad environments, lack of support, etc., can deeply affect mental health. Substance abuse is a way that people cope with the stress that they are feeling. It can worsen mental, physical, and emotional health, not just over time, but immediately as well. Poverty adds to stress as it adds daily pressures that can increase mental health risks through stressful environments. Poverty and substance abuse can raise a risk for serious mental health conditions. This research explores how these factors interact and influence behavior and psychological outcomes. Methods. I did not gather my own data. I used pre-existing data. The data set selection and the source. I looked for a data set that provided data from and about individuals' mental health, behaviors, and environments. The Center for Disease Control and Prevention initiated the Behavioral Risk Factor Surveillance System across the United States in 1984 and has continued this survey until 2023, which is the year I used for my data set as the data was publicly available. When I chose my variables, I chose variables that related to mental health, specifically mental health affecting normal activities, substance usage, and poverty struggles were also other variables I wanted to relate to. To clean my data, which means to turn raw data into graphs, I removed incomplete, duplicated, or inconsistent responses. Then I recoded categorical variables into consistent, analyzable formats. Lastly, I organized the negative factors into a bar graph, which you will later see. In order to analyze my data, I used correlation analysis to identify relationships between poverty, substance abuse, and mental health outcomes. To better visualize my data, I created color-coded charts and graphs to represent patterns and trends. I also highlighted key findings by arranging them into one graph, which is the negative factors bar graph. The next few slides will be my results. As you can see from this slide, a significant portion of respondents have recent or ongoing poor mental health and frequent stress. In fact, nearly one half of the people who responded reported normal activities were hindered by poor mental health. This slide is about substance abuse. As you can see, the frequent alcohol use, which was about 50%, was notably higher than the frequent nicotine use, which is around 30%. These graphs highlight the severity of economic hardship among the population of America in 2023. You can see in this graph on the left that 87% of respondents were unable to pay mortgage, rent, or utility bills in 2023. Majority of the population had enough food, but for around one-fourth of the time, there were people who didn't have enough food. This bar graph shows all the negative factors compiled from all the previous pie charts. As you can see, the highest marker or factor here is the poverty marker, which is the inability to pay their bills within the past year. The discussion, interpreting results. By looking at our negative factors bar graph, you can see a high prevalence of financial instability, like I said before. Substance use shows that a lot more people consumed alcohol than they did nicotine. It also shows that mental health challenges were widespread, seeing as majority of the respondents have recent or ongoing poor mental health and stress. As noted, food scarcity is reported less frequently than all the other issues. The correlation between the variables suggests a cyclical relationship. Individuals facing hardship may turn to substances as a coping mechanism, which further exacerbating stress and mental health issues. Limitations that I faced during this project was that these variables were self-reported, so it can have some biases. A reason for these biases would be underreporting due to lack of awareness of certain issues, along with there were no distinction between severity levels when questioned. So we don't know if the stress or mental health experiences were mild, moderate, or severe. They were all just under one umbrella of whether you had them or whether you didn't. Future directions. Why is this research or data important? It addresses interlinked societal issues. For example, how poverty and substance abuse are connected and related to mental health issues. It provides an overall understanding by connecting variables which were used to underline mechanisms, and it reflects the current state of America, providing a contemporary snapshot of modern health and social struggles. Some things that BRFSS can do to help improve data and help research, such as this, would be to include more localized data. This could mean targeting underdeserved communities. They could also track longitudinal patterns by exploring ways to use data over many years in order to find trends over time. As mentioned before, an improvement in mental health measurement by asking more complex questions to improve the research depth, for example, and like what can be done. Yeah. What can be done to help improve this research in the future? Like mental health can have a broader focus. For example, more detailed coping behaviors and specific mental health conditions. For example, bipolar disorder, depression, or anxiety. By listing these exact disorders and conditions, it can be tracked more and explained better. Incorporating qualitative data which would include focus groups or case studies to have more depth and complexity into this research would help a lot with more projects like this one or just anything in the mental health and behavioral aspect field. Thank you for listening. Any questions?
Yay! Great job, Kushi. And I just wanted to say, I put in the comments that you did a really awesome job taking on this difficult subject, and that you showed really wonderful dedication despite it being a topic outside our main area of expertise. I know both myself and Marin we didn't, it's something that I knew a little bit about, but even when, we couldn't always help you with something, you still stayed really determined and really committed to your original project. And that just shows really wonderful commitment to the ideas and your dedication to understanding these patterns. So I wanted to say well done for that, and I will add your mentor on in a moment.
Thank you.
Okay, and let's see, do we have any questions in the chat? Or from Marin? So I'll ask, I know that this was challenging because we knew less about this subject area. And what helped keep you motivated to stay with the project and to stay with your ideas despite, some of our limitations? What was your driving passion for this project?
Honestly, I think it was just that, my passion. I really wanted to do something relating to like mental health and psychology. So that drive just like kept me going.
That's wonderful. Let's see if I have any other things. And as people might not know, sometimes doing certain types of research that involve human subjects can be really challenging because there's a long process that people need to do that involves institutional review boards for human research subjects, and it can be quite lengthy, and there are a lot of rules around these sorts of data sets. And so it is something that is quite challenging for people to take on, especially when you're coming into ninth grade. So I want to say again, well done.
Thank you.
And we will get to our next student and mentor group. So I am going to introduce Karina, who will be introducing Mina. Welcome, Karina.
Hi, everyone. My name is Karina. I'm a PhD student at the University of Nevada, and my research is in conservation biology. So I was very excited to work with Mina Van Roy, who is interested in studying how humans impact the environment. Mina is from Palo Alto, California, and she expressed to me that she was particularly interested in studying plastic pollution in water bodies, such as the ocean. I had a lot of fun working with Mina to figure out what data was out there that we could use to conduct this research. And I was very compelled by the findings in her research. I was also very impressed with Mina's ability to do all of this data collection and analysis while traveling across the world in Thailand. So with that, I will pass the mic off to Mina.
All right. So taking Karina off. And Mina, just let me know when your slides are ready.
Is it sharing?
Yes, wonderful.
Great.
Okay, and now it is all yours.
Hi, I'm Mina Van Roy, and this summer I conducted a study on how microplastics and marine species richness are related, and I was mentored by Karina. So globally, plastic pollution is a growing problem. As plastic production grows, this plastic needs to be disposed. We have seen much of this end up in the ocean, and it can break down into microplastics. Microplastics range from 5 millimeters, about the size of a pencil eraser, to 1 nanometer, smaller than the width of a strand of human hair. And this pollution is a growing problem affecting both aquatic life and human health. Also, we have seen a significant decrease in global species biodiversity. This has been well documented and could be caused by a variety of factors. As we can see in the graphs on the right on top, it shows the microplastics in the surface of the ocean, how they have increased and how they will continue to increase depending on the plastic pollution we emit. The graph on the bottom shows the decrease in global biodiversity with the Living Planet Index. So, marine wildlife often mistake microplastics for food and consume microplastics. Microplastics attract and carry pollutants in water, as well as release chemicals into the water around them. Lab studies have shown that microplastics and chemicals in plastics may delay an animal's development, cause problems with reproduction, and even make it difficult for them to fight off disease. So this led us to the question, could microplastics impact species levels? Could these levels relate to species richness? Species richness is the number of different species represented in an ecological community, landscape, or region. It is a component of biodiversity, which we used in our study. So our hypothesis was that there will be a negative correlation between microplastics and species richness. This means that as microplastic levels increase, species richness levels decrease. We made this hypothesis because we know that microplastics have negative effects on human and wildlife health. So in our methods, we tried to find data sets which provided location-based data points of biodiversity and data sets which provided location-based data points of ocean surface microplastic concentration, with both being close in collection date. So we used AquaMaps, or Marine Species Richness Database, which is based on plenty of studies and uses a computer model to generate species richness data for oceans across the world. And then we used a study, Egger et al. From 2020, which used Newton trawl deployments, which is a type of net pictured on the right, to collect samples in the ocean and measure microplastic concentration in the Pacific Ocean. For our analysis, we used a generalized linear model with a negative binomial distribution because the dependent variable, species richness, was count-based. So our results indicated that increased microplastic levels correlate with decreased species richness. From the regression analysis, using a negative binomial model, we had a negative slope, which, as we hypothesized, there was a negative correlation. This is represented on the graph by the blue line. Our confidence interval showed that for every 1 unit increase in microplastic concentration, species richness is expected to decrease by between 0.24 and 1.02%. This is shown by the shaded blue area on the graph. So our p-value was 0.00192. This being less than 0.5 indicates 0.05 indicates that our results were statistically significant. However, we also performed a Spearman correlation test, and our results indicated a weak negative correlation, which means that as microplastic levels increase, species values do tend to decrease slightly, but the relationship is not strong. So this is statistically significant, but the correlation is low. This may be because there are many other factors which could impact species richness, including water temperature, salinity, nutrient availability, and coral reefs in certain locations. This would explain our lower correlation value. We also applied these methods to a larger global study, and there was a relationship even with different data ranges. So, what does this mean for conservation? It seems likely that there will be increased plastic production, plastic levels in the ocean, and microplastics in the ocean. Our results indicate that this could have impacts on marine species richness, and we should try and prevent that in any way we can. So one possible source of error was the date of data collection. The microplastics data was collected between 2015 and 2019, while the Aquamaps is based on surveys collected in 2019. This could raise questions about the matching of data points if they are from different dates. However, this was the best available data we had. So some possible future studies would be we could perform a similar study in fresh water, looking at the relation between microplastics in freshwater and freshwater wildlife species biodiversity. Another possible future study would be looking at the mechanism between microplastics and species richness, because we don't exactly know that yet. We do know marine animals may consume microplastics and this can affect their health, but we don't know how this could directly impact biodiversity. So thank you to everyone who's helped me work on this project and made it possible. Thank you.
Yay! So this is a topic near and dear to my heart because I do one-woman beach cleanups and I pick up a lot of plastics. And Nina educated me in that some of the things that I called microplastics were actually kind of macroplastics that will break down eventually into microplastics. But I learned something from you. So, and I want to add in your mentor to see if you have any questions.
Yeah, nice job on your presentation. I think it's really meaningful research that you did. And I know a lot of people are really interested in microplastic research right now. So that was really cool to see. One of my questions for you is, I think you had mentioned to me that in this process, it was your first time using a coding program. We used Program R as an analysis tool and a plotting tool. And I know this is pretty common for doing research in general. So I was wondering, like, for you and your experience, did you think it was helpful, intuitive? Is it something that you think you'll continue to use in the future?
Yeah, I definitely think I would continue to use it in the future. It was like very helpful in tasks that I perceived as would take a lot of time.
Yeah.
To help with that. So I liked it. It felt intuitive.
Yeah, it seemed like you caught on with it pretty well. So that was cool to see. And my other question was, okay, so you sourced your microplastic and aquatic species richness data from public online sources. And this is a skill that I know is like great to have because it's a way a lot of researchers they collect their data that way or they supplement their data that way. So I was wondering, like, if that process went the way you thought it would go, were there any like unexpected challenges to doing that? Or pleasant surprises?
I think I was very pleasantly surprised by NOAA's like full microplastic database, which like listed all the studies which it had. That was very nice. But I think I expected it to be somewhat challenging, and it was a good challenge to try and find nice databases. So that was good.
Yeah, the NOAA database was very cool because it pulled a lot of studies from all around the world together into one place, and I liked that a lot. And I found that the more you look for these online resources, the better you get at finding them. So yeah, nice job on everything.
Yeah, that's an awesome skill set. Wonderful. Thank you so much. I'm going to take Mina off, and I will keep Karina on because the next student is yours as well. So, Rith. And
Hi.
Karina, I'll let you.
Oh, sorry. Thank you. Hi, everyone. In case you're just joining now, my name is Karina. I'm a PhD student at the University of Nevada, and my research is in conservation biology. I was very excited to work with Rith Nguyen, who expressed that they were interested in marine biology and in California native ecosystems. Rith is from the Bay Area in California, and it was really cool to use his local knowledge of wildlife and the environment there to design a research question and to design data collection methods as well. I was very impressed with Rith's ability to go out and collect all of the data that he did, as well as analyze that data in such a short amount of time. And I'm excited for everyone to see the work that he's done. So with that, I will pass the mic off to Rith.
Wonderful. And Rith, just let me know when your presentation is ready to go up. And unmute yourself really quickly. Still quiet. And while we're figuring this out, I'll also say that I was also really impressed by Rith's knowledge of the Bay Area ecosystems, as I also in this area and love going in tide pools. And he was also very, very excited about the native species of snails. And I was inspired by his excitement. So Rith, can you go down to click the icons by yourself in the upper right corner and edit your mic settings?
Can you hear me?
Yes. Yay.
Oh, I'm sorry. I had an external microphone and it was connected to that. But I
Perfect. Okay,
well,
let me put yours up again. Okay, it's all yours.
Thank you so much. Hi, everyone. My name is Rith, and I am a high school sophomore from Wilmerding High School in San Francisco. My study is about the effect of salinity, pH, and temperature on the eastern mud snail in Foster City, California. Okay. I've always been interested in marine life, especially estuary and tidal habitats. I chose to study this eastern mud snail because I was fascinated by how fast they can establish themselves and destroy native species. I was also fascinated by their ability to tolerate different salinities and pH. Though they are tolerant of varying salinities, pH, and water temperature, I wanted to know if specific subgroups within a population, like far from a river, closer to a river, maybe they distribute themselves differently in response to these conditions. So this snail here on the left is the eastern mud snail. It is about 1 inch. It's originally from the East Coast estuaries. It was introduced to the West Coast in the early 1900s along with oyster seed. And I believe this was done accidentally, as now they are present throughout the Bay Area. As you can see that middle photo, all those little dots are snails. This snail eats algae and carrion, and in return, it's eaten by birds and crabs. Because of its estuarine habitat, lives in estuaries and mudflats, it's tolerant of a lot of varying temperatures and salinities. Okay, invasive species. So an invasive species is an exotic species introduced from a different area that this species destroys local ecosystems. Most have little or no natural predators, allowing them to proliferate and outcompete native species. The eastern mud snail is an invasive species, and it's established in the San Francisco Bay, parts of SoCal, also up in Oregon and Washington. So I collected my own data. I was able to pinpoint the population of snails in this area through looking at data from iNaturalist, which is, you can see this map, these two maps here. So iNaturalist is an app where people can post pictures of different species, and other people can confirm and disagree with them. It's really important to help scientists track spreading invasive species or populations of rare or native or endangered species. Each red point on the side represents one submission of the eastern mud snail. When you type in a species name, it just gives you a map of all the observations. And as you can see, there's Foster City, and there is a lot of red markings, meaning there is likely an established population. On the right here, I took some photos of other invasive mollusks I have observed during my trip to Foster City. Foster City, let me tell you a little Foster City. So it's undergone extensive human development over decades, making it an ideal environment for the introduction of various mollusks and other species. And all of these mollusks and other species, I believe most of them come from Asia and the East Coast, and they're all invasive. Okay, more about Foster City. It's a suburb, as you can see on the right. It looks very nice, but it was actually built on a landfill, and it's a man-made island. It's changed since then. As you can see, it's very nice. There are a lot of nice homes and outdoor areas. The ecosystems here are also proliferating. There are a lot of mud flats, salt marshes, which host a variety of animals such as shorebirds, crabs, and other macroinvertebrates. They also serve these estuaries. They serve to protect against floods. I chose to study data here out of all these snail populations because it was the closest to my house, and also they had a nearby river. It's kind of obscured in the photo, but I assumed that there would be lower salinity, which might influence the snail distribution. Okay, my question was to determine if three factors, salinity, pH, and water temperature, play a role on mud snail density. My hypotheses for salinity was that there would be higher densities of snails in higher salinity or waters with higher salinities because the salt helps their cells grow, especially juveniles who are shell and still soft and lacking in calcium. And I read somewhere, I don't think I linked it, but one paper stated that if they don't get enough calcium carbonate, juveniles, they will develop an internal shell, which is basically a massive calcium carbonate inside of the snail, which could lead to death. So it's very important for the snails to get calcium. For temperature, I predicted that there would be low densities of snails in warm pools because they're often warm pools are often exposed during low tide, meaning animals like egrets and shorebirds have easier access to them. Over time, the population might have just died out or they moved away because they knew they were going to get eaten. For pH, I did not think there would be a significant effect pH because the main predators for snails, birds, they don't really have a preference for pH. They mainly rely on sight and sound to hunt, and they forage in pools regardless of pH level. Okay, here are my methods. They're a bit confusing, but I'm going to try to do my best. Okay, so I chose 6 sites. 3 were near the bridge, and 3 were near a river. I'll show you in the next slide. I have a map. I used a random number generator to generate the number of steps I walk on the mud flat and the number of degrees that I turn. At each point that the number generator generated, I placed a 1 foot by 1 foot quadrat, as you can see right here, and I counted the snails in the quadrat, and I also had a little notebook, and I counted for the other animals, invertebrates and animals that I found in the quadrat. Additionally, I measured 3 variables, salinity, water temperature, and pH, using this little device here, as you can see on the right. Okay, here's a map if that was a little confusing. So here's Foster City. There are 2 main habitats where I collect data from. The first is near the bridge. I believe it's the San Mateo Hayward Bridge that starts in Foster City and it leads to the East Bay. And if you can see the middle one, I collected 3 samples there. And then down at the bottom of the first picture, this other site is closer to a river, which is actually a slough, which is brackish water. Okay, so to see if my results were statistically significant, I used a linear regression. I used a p-value of less than 0.05 as a cutoff. The value of 0.05 right here, the p-value for salinity is greater than my cutoff number, meaning that these results may be due to chance. The null hypothesis is a hypothesis where there's no genuine connection between the variables. The results here for salinity show no significant relationship, meaning cannot reject the null hypothesis. And also you can see there's a negative slope here. The results do show a negative trend. As salinity gets higher, the average snails decrease. I found these results very interesting. It was the opposite of what I predicted. I predicted that there would be more snails and higher salinities because it helps their shells grow, and it turned out there were less snails as salinity increased, which was fascinating. Temperature, there is a similar negative trend here and a high p-value of 0.8. And as you can see, another negative trend. And that was not what I predicted. Moving on to pH, again, a similar trend, relatively high p-values above 0.05. Pretty high p-value. suggesting that these results are likely to be due to random chance. In addition to these three variables, I thought it would be interesting to graph the site number. I sampled 6 sites, 3 from a bridge and 3 from a river, just again. I found actually the p-value is less than 0.05, which means there's actually would be a significant relationship between the snail count and the site location, meaning that there might be a factor that I didn't really think about in the environment, like maybe the type of substrate, the increase in bird species in one area that could be affecting the snail densities. I came up with some possible reasons for this. The first location in the bridge had a lot of litter. It was near a bridge, and I was thinking maybe there could be food in the litter, and they could be drawn to the food, and it's affected by humans, so people might accidentally like drop litter there, and then they would be attracted to it. The second location near the river, I noticed that there were a lot more animals and different plants and birds in that area, more biodiversity, less litter, and it was near the suburbs, which could, I was thinking that the increase of predators in that area might have driven the snails away. So this also brings up the topic of what an indicator species is. So an indicator species is a species that they're species that are used to measure how healthy an ecosystem is. There are some species that are persistent even when the water quality is very low, and there are some that are present when the water quality is really clean. And I was thinking that the snails could be an indicator species because in the first 3 sites, there were a lot of litter and a lot of snails. And then in the last 3 sites, there were not a lot of litter, more predators, but there were lower amounts of snails. Like I mentioned before, there is a varying biodiversity between the 2 sites. I used iNaturalist to track how many animal species and plant species I found. In the first site, there were 11 animal species and 1 algae species. There are a lot more invertebrates, actually, like crustaceans, snails, mollusks, etc. The second site, there are 4 animal species, and I'm pretty sure 2 of them were snails, and then the rest of them were birds, and then there were 4 plant species. And my pictures show invasive. Here are invasive eastern mud snails on a Gatorade bottle. In the first, the first 3 sites showing the pollution, and then the native California horned snails, which were actually driven out by the eastern mud snails. And these mud snails were observed in the second site. Okay, so what impact do these results have on conservation? One reason why these results are important is because they can inform companies who want to build houses or want to build buildings along estuaries about the distinct and important habitats that occur within a stretch of an area. And I believe between my first site and second site, it was only like a couple 100 feet, but it was a drastically different habitat. And this can help companies choose their build sites wisely without disturbing wildlife as much. Some future studies idea would be, one future study would be testing if the mud density has an effect on the snail density, because I noticed in the second site, there was a lot more clay and it was much thicker. In the first site, it was silty and it was pretty hard to walk on without sinking into the mud. I was also thinking about studying, analyzing the predators in the area. As I mentioned before, the second site exhibited greater biodiversity among birds, while the first site exhibited greater biodiversity among macroinvertebrates. An initial idea I had for this study was to compare the density of these invasive snails to the California horned snails, which I mentioned. During the spring months, they do occur in the same area, but I read somewhere during the summer, the eastern mud snails drive the California horned snails up to higher intertidal areas, which means they don't coexist. So a future study could be comparing the densities of both the invasive and native snails when they coexist in the same area. Some sources of error could be, number one, not collecting enough data among different habitats. I was thinking about in the beginning collecting about 15 data points, but I only ended up collecting 6. I do plan to collect some more data, which might help me out. Number two, the sites were drastically far apart. If I go back to the map, you can see there's just a clump of sites near the bridge and a clump of the sites near the river, which definitely could affect the results. Also, the water pH and salinity samples might not be representative of the population. I took my gadget and I sampled different pools, but I didn't really have a method in sampling them. So if I had some random number generator give me amount of feet I walk and sample for that, it might have been representative. I also didn't survey all types of mud snail habitat. I only surveyed 2 types of habitat. So if I had surveyed different habitats like tide pools, maybe I wouldn't gotten more data to interpret. So in conclusion, I did not find a significant relationship between the average mud snail count and pH, water salinity, and temperature. However, there's a noticeable trend related to site location. And some possible explanations could be there are different factors that are affecting the results, which could be leading to the relationships, and also substrate type, vegetation, and human disturbance may affect the results as well. Thank you so much. I would like to thank Courtney Sanchez, my mentor, Leslie and Megan, Headwaters staff, and of course, my parents and sister for helping me collect data and supporting me. Thank you.
Yay! I'm going to add your mentor on.
Hey, great talk. I thought your results were surprising with the water quality maybe being less important than other factors, although we know your results might change a little bit with additional data points. But yeah, it's very interesting information for conservation management. So I was wondering, how did you first become aware of the eastern mud snail problem in Foster City?
Yeah, so I actually first saw mud snails when I was in Maine, and I was looking at the population in Maine, and then I scrolled out of the United States, and I saw some on the West Coast, and I was like, oh no, where are these mud snails? Why are they spreading so fast? So I zoomed in around the SF Bay Area, and I found this population, and I was like, oh my God, why are they spreading so fast? This is not good, and I decided I wanted to do an experiment on them.
Interesting. Yeah, I didn't know that you had seen them kind of more in their maybe native habitat as well. So yeah, the invasive species problem is big right now. So it was cool to see what you found. And my other question was, so collecting your own field data is impressive because there's a lot of logistics that you have to figure out, like the tools that you need to get, figuring out survey locations, a consistent data protocol. I mean, you had to even work with the tide schedule. So I was wondering, like, what was your experience collecting your own data? Was it challenging? Was it something that you think was worth doing?
Yeah, so I definitely thought it was challenging going out in the field, especially because we had a short amount of time to record the data. But it was pretty fun and it was rewarding after I collected the data. And also, I love being out in nature, so it was great to be out in the estuary and observing the wildlife.
Yeah, I thought it was great because I think you got a lot of insights too that you wouldn't have if it was someone else's data set. Like you noticed things that might influence like the snails based off of their location because you got to see the soil that they were on and the predators that were around. So I thought that was cool.
Yeah, I had a question. So I also have to say it's definitely challenging to navigate those mud flats. I spend a lot of time recreationally out in those, and like you do really have to pay a lot of attention to the tides, and you I've spent a lot of time being stuck, and then it also, I don't know if over there I'm on the opposite side, it's like has quite a strong smell, the mud as well. But so I was wondering when we talked also in your practice about the connection between the snails and the sites. I was wondering if any of the snails in your study were filter feeders, and I know that there's some species like larval abalone can have negative impacts if there's more sediment in the water. And I was wondering if you noticed any difference in the water quality in these different locations other than what you measured, and if you think that what was used as landfill in Foster City could have any impact on the ecosystem, because a lot of the bay is landfill.
Yeah, so I took pictures of each site, and I believe in the first site, the waves were like more rough. I didn't measure any other variables, but I realized like the waves were rough. And then in the second site, I noticed they were like calmer. Maybe they could preference the higher waves for some reason. And your question about the filter feeders, I believe I don't think they're filter feeders, but they do eat algae and carrion.
Mm-hmm.
But I do think, yeah, the waves and also like just the general area, landfill area, okay, and human impact could have definitely had an impact on the snails.
Yeah. Awesome. All right. Do you have any more questions, Karina?
No, I think that's it for me.
Okay, so great job again, and I'm going to get the next person ready. So next I'd like to introduce Megan Joyce, and because Megan Joyce has done some mentoring of me, I want to thank her for being a good mentor as I've tried to learn the nuances of some of these systems like Google Classroom and StreamYard, and that she's helped immensely in teaching the communications course this summer and also helping me navigate these new programs, which I really panicked about, especially StreamYard. And so I appreciate your skill in using these. And after I have used it, I see how it can provide a really nice, clean experience. But Megan definitely has a lot of years of experience teaching this class and doing communications. And so I will introduce her to introduce her student.
Thanks, Leslie. And it's been awesome to see all of the presentations so far. Everyone's doing a fantastic job. And as Leslie mentioned, I'm Megan. I sometimes teach the course. I have a background in communications and wildlife conservation. And so I was really excited this summer to work with Lila. And I won't give too much away because she's going to tell you all about it. But I was super impressed with her rather complicated mechanism to collect her data. And she collected tons of data and got some really interesting results. And so one of the challenges, which I think she'll mention, but just the timing of her project was really specific on when she needed to collect her data. And so she troubleshooted a bunch and collected a ton of data, and I'm really excited to see her full presentation and results.
And just a moment, I'll take us both off. And it is all yours.
Thank you. Hello, everyone. My name is Lila Fortner. I am a rising senior at Justin Siena High School here in the Bay Area. And my project was on osprey behavioral responses to fireworks, specifically fireworks on the 4th of July. So what really got me into this project was where I live on my island. There's a couple of osprey nests, I think about 3 that are on the island. And one of the coolest features about one of them is that we have an osprey camera on one of our nests. So I was thinking about what I could do to involve these birds that are really cool and you hear them and see them all the time in your daily life here on the island. So as I started going into my research background about fireworks and bird, osprey, animal responses to them, I actually found surprisingly little about osprey specifically. But I did find a lot of data about how domesticated animals are often spooked or very anxious when it comes to these times of years, like New Year's or 4th of July, where a high firework activity is very common. And also there are some other animal studies, such as zoo animals like horses or other land mammals that were involved, but not many involved birds of prey or raptors in general. So I went a little bit deeper into what I could find about ospreys' general demeanors that could give me any sort of hints of how they would react to such loud, abrasive sounds. And in I found a couple different studies. A couple of them were human disturbance studies, and a couple were just general osprey behavioral studies. I found that ospreys are a naturally aggressive species. They have been shown to be disturbed by human disturbances, whether that be a wind farm, jet airplane noise, or even just another bird or a drone or a human approaching their nest. I took a lot of methods away from them, such as you will see later in my method slides, different ways of categorizing behaviors and disturbances. I had eventually had to create a numerical system, as you will see. But I learned a lot from those studies, and it made me very excited to see what I would learn from this study. So this leads me to my question. So my question was pretty simple. Is there a correlation between firework activity and osprey nesting behavior? Just trying to get a general sense of what it would be like, what these birds might behave like when presented with this very new, very human-made disturbance. And my hypothesis was that disturbance-related osprey behavior will increase in frequency during times of firework activity as opposed to times of non-firework activity. So I used controls of no firework nights, and as I labeled them, and I used experimentals of firework nights that was on the 4th of July, 4th or 3rd of July, and led to that's what I used for experimental. So my methods, this was, as my mentor Megan said, this was probably the biggest chunk and hurdle of my whole project. I had to use software to capture live streams of osprey camera nests because I cannot be everywhere at once and I could not watch that many different nests. So I had to use software to capture live streams, but in order for the live streams to even be something I could use, they had to meet some very specific criteria. Some of those being that they had to have some sort of night vision, as you can see on this bottom photo, which is one of my locations, which was Longmont, Colorado. They had to have night vision so I could see the birds when I was at night, because if I couldn't see the birds, then I couldn't see what they were doing. It had to have audio so I could hear the birds calling, because alarm calling is one of the behaviors that I was looking for. So I had to be able to hear what the birds were going to be vocalizing. It also had to have some sort of 12-hour memory so I could go back in case my recording didn't work and watch it over again, just to have a fail-safe, really. It had to be on YouTube because the software I was using did not allow for any other type of link. I found a couple that I was able to just simply screen record, but those became a little bit more complicated. And then finally, they had to have a fireworks show on the 3rd or 4th of July. And that, luckily, every single one I found was able to work out that way. So there was a lot of limitations and criteria that I had to use, but I found about 7 locations. There was actually originally 10, but about 3 of them did not actually work out with the recordings. Still do not know completely why, but I ended up getting 1 to 2 controls of each nest. I only used 1 for my experiment though, and I got 1 experimental night, which was the most important night to get. That was a very stressful time to get it, as Megan said. I was in a very time crunched moment to get everything. So that either happened on the 3rd or 4th of July to capture all of the festivity activity, especially fireworks. So, and in addition to just making sure it fit the criteria, I made sure to get some other information. I recorded whether there was chick presence and the number of chicks, because as you can see on this bottom photo, there was definitely 2 chicks in this one, and that was a very common occurrence in many of the nests I found. But there was also some nests like this top one where this is in Hellgate, Montana, where there was no chicks. It was what they would call a failed nest, and oftentimes the bird was not there, and it turned out that they were not there the entire time I was recording. So I recorded that to see just general bird behavior in relation to the chicks. I recorded the locations, so I ended up getting 7 locations, which were Hellgate Canyon, Montana, Mare Island in California, where I live, Longmont, Colorado, Lowe's, Delaware, Independence, Oregon, Narragansett, Rhode Island, and in Mooresville, North Carolina. So it was a good spread. Most of them were on the East Coast, but I found a couple on the West Coast as well. I also recorded where the fireworks would be taking place, so I recorded how far away the fireworks would be to see if there was any sort of discrepancy in the responses of the birds, depending on how far away the fireworks might be, because sound eventually just does not travel as far. So the furthest I got was about 6 miles, and the closest, as you will see in one of the photos I have, there was literally fireworks about 50 feet in front of the nest, which was a little bit disturbing. And then finally, I also collected how long the nest had been there for. The shortest amount of time was about 3 years, I want to say, and the longest was almost about 15 to 16 years of the birds nesting there. And then to continue on my methods, I ended up getting 1-hour recordings, and this was so that I could get all the fireworks shows were about 20 minutes long, so I would get 20 minutes before, which I called the pre-fireworks stage. There was the fireworks stage, which was 20 minutes long, and then I got a post-fireworks stage of also 20 minutes. So to ensure that I got a baseline of what they were doing right before the fireworks started, what they were doing while the fireworks were going, and then also what their immediate reactions after the fireworks were over. So, and in order to get all of my data collected, I created a rather extensive chart that included where the nest was, the date that I had the recording, and the timestamps of each of the individual behaviors, what stage it was, so whether it was that pre-fireworks stage, that fireworks stage, or that post-fireworks stage, just to make sure I knew when exactly in the recording it would be. I also had, were likely my 2 most important categories and rows, that I had a disturbance index and a behavior index. There's a slight typo on this slide where the behavior index was actually from 1 to 4, not from 1 to 5, but those were probably my 2 most important, because that would be what I was basing my data off of. And that is something that I took from the literature that I read, because oftentimes in behavioral studies, they use a numerical categories to give a numerical way to analyze what was happening in the field, because it's hard to analyze any sort of behavior in some ways. And then also I used shorthand to write down any behavior in relation to disturbance. So you can see in this bottom right, you can see what we called the codes. So there were 3-letter codes to determine what the behavior was without having to write it out completely and try to hopefully shorthand some of my data. They ended up being very helpful. We ended up seeing more of some and less of others. Here, right below my writing, you can see my disturbance index and my behavior index. So the disturbance index, we were just quantifying for the disturbance of fireworks. There was some boat noise, but the birds generally seemed habituated to that. So most of it was just firework noise, and it went on a scale from 1 to 5. 1 being there was absolutely no disturbance at all, in what way. And 5 was as shown in this one photo that I have. This is from Independence, Oregon. You can see the bird leaving her nest there, leaving her chicks behind as the fireworks go off literally right in front. And that was the level 5 disturbance as I quantified it. And then you can also see my behavior index below. You can see that I associated certain behaviors with certain levels of disturbance. One being that their regular behavior, resumption of regular behavior, or no bird, because I did have some nests where there was no birds in the nest. And 4 meaning that the bird had either left the nest, the chick was having a response, or the bird was gone for an extended period of time, which didn't end up happening in that Oregon nest. So overall, these results took, I ended up getting quite a lot of footage to go through, and there was a range of responses and disturbances. Some places you could hear the fireworks better than others. Other places the birds seemed to have a bigger effect, and we will see on my results slide. This is what happens. So I used an ANOVA test to analyze my data, and I got a p-value of less than 0.01, which means that these are statistically significant in that these groups are definitely different. So this is all my data, including my controls. I eventually separated my controls and my experimentals just to see if there was any differences. That will be on the next slide. But out of my 283 points of data across my experimental and control data sets, this is what I got. As you can see, there was not that many events where the disturbance index was 5 at the greatest level of disturbance, and that only happened at about 1 nest. But there you can see that whenever it was, they were definitely it was a big response from the bird, let's just say that. So typically the bird would leave the nest. It was gone for about 15 minutes, leaving her chicks behind, which from what I could tell from studies before, they rarely ever do that just to make sure that their chicks fully get to adulthood. But you can also see that there's some big differences along most of my data. There's a couple outlying points where, like, for example, when the disturbance index was 1, there was one event where the bird was very agitated at a behavior index of 4, which is the biggest level of disturbance I could see. So this was my general data. So these groups 1 and 2, traditionally, as I ran multiple tests, my post hoc Tukey tests, they showed those were not very statistically different, which means that to a certain degree that the birds were not very affected by certain levels of disturbance. It was when you got to groups 3 and 4, which also showed to be very similar in many different tests, that the birds started being a little bit more disturbed by what was going on. Then obviously when the disturbance index was 5, it was a very big reaction from the bird. And one thing that I found very interesting was the fact that when the disturbance index was 3, there was the most different range of behavior indexes from the birds. So you can see there's some that are where the birds were very agitated with a behavior index of 4. There was many where they were not disturbed at all, and it's at a 1, and there's plenty in between. So that was one thing I was very interested about, that there was, in this middle range of disturbances, there was not a lot of fluctuation between what the birds were doing. So here you can see kind of my control and experimental data. My control, I actually was surprised to see the fact that there was some even disturbance indexes of 4. That was likely due to the fact that during some controls, I did my controls leading up to the week of the 4th of July, and there was a couple premature fireworks that I could not control for, but we worked with it anyway. But I could tell that for the most part, those control lines were very calm, no disturbances. The bird, for the most part, did everything that was normal for a bird to do. And then my experimental data only, I see something very similar to my control and experimental data. All of my tests came out the same. I did ANOVA and post hoc Tukey tests still, and they showed me that there was still statistical significance with a p-value of less than 0.01, that there was still statistical significance between all these groups, meaning that as the disturbance index went up, the birds also became more and more agitated until you got up to the disturbance index of 5, where they were definitely disturbed at any point in time. So what does this mean for my study and my research? So this means that the ospreys were showing signs of disturbance to firework activity, which I was very excited to see, because it proved my hypothesis correct that there is an increase in frequency in disturbed behavior of ospreys when there is more firework activity. This makes sense given the context of how protective and alert ospreys have proven to be over their young, because oftentimes the child rearing or the chick rearing process of ospreys' lives is very volatile. Chicks, they can lose chicks at many different times, so they're often very protective and therefore very alert and very aware of what's going on around them. Couple trends that I did see in the data was that typically when the fireworks show first started, the birds seemed a lot more agitated than, say, near the 20-minute mark of the fireworks show where the fireworks were starting to end. The birds started to calm down a bit more, just kind of accept that it was happening and that it was just something that wasn't going to harm them or their young. So I definitely saw a bit of a petering off near the end of the studies. And then I also saw that in general, I don't have much data to back this up, but as a general trend, I saw that nests that had been in those locations for a bit longer seemed, the birds, especially the adults, seem to not be as affected by the fireworks, which I presume means that they've been through multiple New Year's days and eves and multiple 4th of Julys where fireworks have gone off, and they know that it's not something that's going to harm them or their young. And also finally, that as a general trend, the chicks did not seem to care very much. Many of them just slept through the entire fireworks show, even when their parents left the nest. So it seems like the chicks do not seem to have, they're not much affected by these giant displays of man-made noise. There was also a couple of discrepancies and possible sources of bias or something in my data. There was 2 nests that were, unfortunately, there was no chicks. That meant that there was no bird in the nest at the times that I was observing the nest. So therefore, I did not get a very good grasp of their behavior. They would show up their nest periodically, but unfortunately, I did not see them in the nest while I was doing my recordings, because my recordings were so time sensitive. I could only do 1 hour. I couldn't really shift it. I had to make sure that my control was recorded at the same time as my experimental recording would be recorded. And so it just led to a couple of times where the bird was not there. And so that could have lead my data to one way or the other. And there was also a problem was I was very much limited by the number of cameras with the proper criteria. In general, there's already a limited number of cameras in the United States that are on osprey nests, and also in addition to that, there was even a further limitation by the fact that not all of them met my criteria. There was a lot that I really liked the location of, everything worked except that I couldn't see them during the night, or I couldn't hear them, or something else was off about the camera that just made it unusable. So where does this lead in terms of everything else? Well, the birds definitely do show signs of disturbance, but a couple of questions I would have for the future maybe is, do the birds who live in maybe very noisy, very loud areas, such as possibly thunderstorm-prone areas or areas with loud freeways or boat noise, are they more accustomed to loud noises such as fireworks than maybe other birds? And also in my study, I saw a wide range of chick age. There were some that were very young, still very much fledglings, and there was others that the day after I collected all my data, I went back just to check on some of the nests out of interest, and one of the chicks had finally learned how to fly. So there was a wide range, so I'm curious to see if maybe when the range of age in the chicks causes a different reaction. But those are all questions for the future that I could not have time or resources to fully flush out in this research. But I did discover that the ospreys do show signs of disturbance due to firework activity. So here's some acknowledgements plus some of my references. I could not fit all of them on here. But thank you to Megan, my mentor, who helped me through this process. The amount of times that I thought I had something under control, then something went haywire, and I had to check in with her and make sure what I was doing was all right, and then I was getting on the right track with everything I was doing. Thank you so much for helping me through all that and through all of our Zoom calls that we did. And thank you also to Leslie for teaching the class in such a great way to help me understand everything that I was learning, especially maybe all the new stuff about statistical analysis and some of the specific tests that were being done. You guys both helped me through this process a lot and answered any questions that I had in a great way. And also thank you to my family for watching in some of the footage that I collected, because it turned out to be almost 21 hours of footage that I could not get through in time because it took longer than an hour to collect all the data. So thank you to especially my mom and dad for taking time out of their day to watch a video or two. Meant a lot to me that you guys would do that for me. And then also for my dad in particular, who essentially created the software that I used to record all the live streams. Thank you so much for putting all the time and effort into that. It really, I don't think I could have really done my research without all of that software that you created. So thank you. And that's the end of my presentation. So thank you so much for listening, and any questions?
Awesome job. I'm going to add Megan back on here.
Yeah, fantastic. I'd love to start with just a first question for you, which is not about any of your particular collection or anything, but what was the most surprising part to you of like the whole process? It could be in your results, but it could also just be conducting this research on these ospreys that aren't the ones that live right next to your house that you could just kind of like go out and look at.
Yeah, so there was definitely a couple of surprising things. Some of them seemed a little bit more accustomed to certain sounds than others, like boat noise. Ospreys live by the water, so they always typically they have some sort of boat or water activity going on. Some seemed a little bit more twitchy to that than others. And in general, I also really love to see their everyday behavior, such as their feeding or in many cases, going to the bathroom, especially the chicks who cannot fly. So that was very fun to see. And then in terms of like watching them and for their behavior, I was very interested to see the range of behaviors. I saw something different from nest to nest to nest. I don't think any two nests were the same, and that was very fun to see because I thought that they would all be very uniform.
Yeah, that's really interesting. And is my other question for you just on, you partially mentioned that you didn't have a ton of nests, right, that fit your criteria, but we ended up having a ton of data points for your research. So what was the impact of like that spread of your data on your results?
Yeah, so I would have liked to get a couple more nests. Like I said, I think I had about 10 in total that would have worked if something hadn't crashed the night of doing it. But I think definitely seeing just how specific birds in specific regions of the world kind of reacted to these firework disturbances. I mean, I'm grateful I got the amount of data that I did. I did not expect to get as much as I did. But I definitely think that it helped provide at least a decent sample size and allowed me to make a decent inference for the rest of the osprey population.
Yeah, definitely. And I liked your note at the end too about just how some of the ospreys, the chicks, are at all different ages. And you mentioned earlier that the parents are often super protective over the chicks. And so figuring out some way to kind of test that in the future might be really interesting too.
Yes.
Yeah, I was going to say it would be interesting to see if the response was different between first-time parents and parents that had had a lot of successful fledglings. And I also would be really curious, do you think that the impact is just the noise, or do you think the light part is also impactful?
I think there's definitely a factor that is the light part, because I couldn't always see the fireworks that were happening. Oftentimes I just had to listen to them. But when I could see them and they seemed to be somewhat close to the nest, the birds did seem to have a bigger reaction, maybe shifted around the nest more. One even left the nest for 15 minutes, as I mentioned. So I definitely think the light is a bit startling to them, especially since it's happening at night and they're used to night being completely dark. So I think a combination of how loud it was and how bright it was caused a lot of different behaviors in what they did.
Okay. Awesome. So this was our last presentation of the night, and I don't see any more comments. Megan, do you have any other questions?
No, none for me. Awesome job.
Okay.
Thank you so much.
So, and thank you so much, Lila. And I learned, I think that there's an osprey nest that's on the other side of the Richmond Bridge that is often over kind of by Point Molate. There's a couple different ones. And so I'm really curious now if they have cameras associated with them, because there is some of the frontage that's kind of protected. And that would be interesting. But thank you so much. And I'm going to take you off. And thank you again, Megan. And with that, for those of you who are still here, thank you so much for watching our presentations. You can watch them again on Headwaters YouTube channel where they are recorded. We also will be finishing the students' manuscripts, and you can stay tuned to be able to read those. Thank you again, and have a wonderful night.